A very good afternoon to one and all present here. On this occasion, I request our director to kindly invite the chief guest and other dignitaries to adorn the dais. Chief guest of today's function, Professor Dr. Stefan Grill, Director, Max Planck Institute of Molecular Cell Biology and Genetics, Germany, Dr. Anandavalli, Director, CSIR SCRC and Coordinating Director, CMC, Dr. Anup, Dr. Saptarshi Sasmal, Dr. Hari Krishna, Chief Scientist, CSIR SCRC, other scientists, invitees, colleagues, and friends. I welcome you all to one of the main science festivals of India, National Science Day. We shall now begin the function with an invocation. I request all of you to please rise.
நீராறும் கடலுடுத்த நிலமடந்தை கெழிலொழுகும் சீராறும் வதனமென திகழ்வரத கண்டனிதில் எக்கணமும் அதில் சிறந்த திராவிடனல் திருநாடும் தக்க சிறு திரை முதலும் தரித்தனரும் திலகமுமே திலக வாசனை போல் அனைத்துலகும் இன்பமுற எத்திசையும் புகழ் மணக்க இருந்த பெரும் தமிழனங்கே தமிழனங்கே உன் சீரிழமை திறம் வியந்து செயல் மறந்து வாழ்த்துதுமே வாழ்த்துதுமே வாழ்த்துதுமே.
In the year 1986, the Government of India decided to designate February 28th as National Science Day to commemorate Professor C.V. Raman for his legacy and discovery of the Raman effect. In an effort to popularize the benefits of scientific knowledge and accelerate the pace of development in science and technology, this day is celebrated as National Science Day all over India. The theme for Science Day this year is Youth Empowerment in Science and Innovation, aligning with India's vision for development. Today, we have with us world-renowned researcher, Professor Dr. Stefan Grill, Director, Max Planck Institute of Molecular Cell Biology and Genetics, Germany, as a chief guest. To extend our heartfelt welcome to him, may I now invite our director to honor our chief guest with a shawl. I request our director, Dr. Anandavalli, to begin today's function with a welcome address.
Honorable Chief Guest of today's function, Professor Stefan Grill, Director, Max Planck Institute of Molecular Cell Biology and Genetics, Germany, Dr. Hari Krishna, Dr. Saptarshi, and Dr. Anup, guests, my dear brothers and sisters of this campus, friends from media, Vanakkam and Namaskar. So today we have gathered here to celebrate National Science Day, which falls on February 28, a day that not only commemorates the discovery of Raman effect by Sir C.V. Raman, but also serves as a powerful reminder of the transformative impact of science and technology on our lives and society as a whole. Science is just not a subject, it is a phenomenon around us. Science is an endless adventure that never ends. There is always something that will keep you alive and excited throughout your life. It was on this same day that the great Indian physicist Sir Chandrasekhara Venkata Raman made to the world his greatest invention, the Raman effect, in the year 1928. He was awarded an honor in the year 1930 with the Nobel Prize in the Physics for his great success in the field of science in India. In 1986, Government of India announced 28 February as National Science Day. On this day, we show our dignity and respect to the famous Indian physicist to mark his discovery of the Raman effect. Sir C.V. Raman was born on 7th of November 1888 at Tiruchirappalli, Tamil Nadu. His father was a lecturer in physics and maths. Sir C.V. Raman worked from 1907 to 1933 in Kolkata at the Indian Association for the Cultivation of Science, doing research on so many topics in physics, of which Raman effect became his great success. His discovery marked a notable achievement in the Indian history. So science affects us all every day of the year, from the moment we wake up all day long and through the night. Day starts with the digital alarm clock for some of us, the weather report, the asphalt on which we drive, the vehicle we ride, our food and medicine, our communication devices such as mobile phone, and the light that we turn off at the end of the day have all been brought to us with technology enabled by science. The main objective of celebrating the National Science Day is to bring awareness to people about the importance of scientific applications in their daily life and to encourage them by popularizing science and technology. Science starts with asking questions and being curious about things happening around us. National Science Day paves way, a platform that allows the scientists and the experts from any particular field to discuss issues and new technologies trending in that field. Also, the celebration is aimed at giving an opportunity for the younger generation to display their activities, efforts, and achievements in the field of science for human welfare. Science is a methodological process, as we all know, extracting true facts from any given thought by adhering to a set of rules known as methodology. So, as Thiruvalluvar says, எப்பொருள் எத்தன்மையதாயினும் அப்பொருள் மெய்ப்பொருள் காண்பதறிவது, which means that analyzing the information, understanding the meaning, and accepting the fact will help one to gain profound knowledge. Hence, true knowledge is the ability to understand the truth behind people, things, and events. Science includes observation, as we all know, evidence, and experiment, re-examination, analysis, verification, and review of the results. So, as our former beloved President Dr. APJ Abdul Kalam mentions, science is a beautiful gift to humanity. We should not distort it. As science plays an ever-increasing role, however, scientists must remain true to the fundamental principles of objectivity and impartiality. In some cases, research and monitoring programs often offer the warning to the government officials and to the public of an emerging problem. So science must respond to societal needs and global challenges. Understanding the science behind major global challenges such as climate change, ocean health, biodiversity loss, and freshwater security is essential to overcome these challenges. Every year, we celebrate National Science Day with a new theme to promote a special message. The theme for this year is Empowering Indian Youth for Global Leadership in Science and Innovation for Vikshit Bharat. And this theme underscores the pivotal role of India's youth in steering the nation towards becoming Vikshit Bharat, or developed India. By fostering scientific acumen and innovative thinking among young Indians, the country aims to position them as global leaders in science and technology. This empowerment is envisioned to drive socio-economic growth, address pressing challenges, and contribute to sustainable development. There are several strategies that can be implemented to achieve this, actually realize this vision. Educational reforms have to be brought up, research opportunities and innovation ecosystem has to be built, and mentorship programs. And science has no boundaries, as we all know, and unites people from diverse backgrounds and in the pursuit of knowledge and understanding. From exploration of the outer space to the study of microscopic organisms, from the development of life-saving medicines to the creation of sustainable energy solutions, science touches every aspect of our life and holds the key to the addressing some of the most pressing challenges that humanity faces. And coming to CSIR, we all know that CSIR is contributing to the nation through its innovativeness, and CSIR covers a wide spectrum of science and technology from oceanography, geophysics, environmental science, and also provides many areas concerning societal efforts, environmental health, shelter, drinking water, housing, and whatnot. And coming to CSIR Structural Engineering Research Center, I'll just scan through these slides so that what we are doing, we'll just try to understand. So this particular laboratory was established in 1965 with the aim for self-sufficiency in structural engineering, and it was in this campus by the CSIR governing body. It was 14th January they decided to pursue this, and then it came into existence at Roorkee. In 1969 it came into this campus. And as this uniqueness of this laboratory is that around 80 scientists, 80 to 85 scientists, all working basically belonging to structural engineers, and in the world I think this is the only institute where such a concentrated number of structural engineers are working. So we address different aspects of structural engineering, and we have organized ourselves in 6 different themes. One is structural health monitoring. As we do our health checkup, we also have to monitor the structures and to address the distress. That is one thrust area where we focus. The other one is disaster mitigation, both man-made as well as natural disasters, and an integrated approach for preparedness, including preparedness, how we'll be addressing. And as we all know, natural materials are depleting, so how to address that, and also to take circular economy from the construction demolition waste is the most important waste that has to be addressed. And the thrust area on advanced materials for sustainable structures addresses this, and we are also having special multifunctional structures which caters to different things. And energy and infrastructure and offshore structure is other areas, keeping in view the near future demands. And this is our infrastructure as a whole, and many of you might be knowing. So those who are new to this institute, as well as the occasion demands, I'm just scanning through that. Different aspects, as I have told, from the materials to the analysis design, as well as load estimation and monitoring. All these are covered under 10 different laboratories in this particular institute. And these are some of the few initial installations in this particular campus, which our own technology or our own design has been adopted. And finally, we take it outside and implement in the field also. It's like a demo kind of thing, but lifetime structures. These are all existing, which was built in 1965 or 67. And each of the building has one particular aspect of structural design or something incorporated in that. And these are all like a funicular shell roof. And this is adapted in one of the buildings here, and later it was used by residential quarters at BHEL Tiruchirappalli. And cold deform bars, which was being imported those times, and we have developed the indigenous technology, for which we have also received the award for that. And using that, our own heavy testing laboratory was built. This particular cold deform bars were used, and later it was also taken commercially by the Tata Iron and Steel Company. And brick shell roof is one particular very interesting aspect where the bricks are assembled as a shell roof structure. And these are some other structures where hemispherical raised domes and other things which was patented earlier, and it was adopted in this campus and later taken outside also. And first 2 decades we were on the advanced materials and innovation structural designs, where the fly ash utilization, a waste material which was utilized effectively, and also pre-stressed concrete sleeper where wooden sleepers in the railway lines were replaced with this particular technology, and prefabrication is another area where we have been working. And in the next 2 decades on the societal development and national building capacity, various aspects have been taken forward. And in the next decade, interdisciplinary research, where most of the SHM, structural health monitoring and materials aspects we have been working on. And recent innovations where we have used the acoustic emission and advanced non-destructive techniques has been adopted in one of the important irrigation project. It's a major dam in Andhra Pradesh where this particular technology was adopted to give them the assure the safety of the dam as well as integrity and other things. And towards strategic sector also our institute is being contributing various aspects including fuel drop tank as well as shutdown rods. Another, and in man-made disasters, this is again a full-scale testing of explosive storage structure, and we have designed and we have also developed a new system to address that. An emergency retrieval system, this is also indigenous development which has enabled us to, this particular system is 40% less cost than the imported one, and this is being implemented right now. And renewable energy, as the demand goes, we are also working on different aspects, solar farms as well as wind turbines. And these are some of the latest. So our focus is on cyber physical systems in future and also multi-hazard design and carbon sink materials. These are all different areas which we are trying to work on, do our research, contribute to the society. Bio-inspired engineering may be some of the areas where mutual interest will be there between the institutes. And there is another, apart from ACRC, this campus houses a CSR Madras complex where 5 different laboratories are there, which with the aim of promoting interdisciplinary research, this concept of multi-labs, a complex was created in this particular campus, and each one has got their own unique impact. One is from Central Electrochemical, where lithium-ion battery pilot scale demonstration is there, and 1,000 cells per day. And also they have used fuel cell technology, and they have also demonstrated the prototype. And coming to CSIO, Energy Management System, this is a scientific instrument organization, and also biosensors using nanomaterials. These are all few of the developments which is happening in this campus, and the biomedical imaging platform, all these are being done here. And coming to see NML, National Metallurgical Laboratory of the local center, they are working on the column flotation technique, and also they have developed different eco-friendly reagents, and these are also pilot scale, as well as they have also transferred the technologies to multiple industries. And CD unit, various aspects like how to identify the defects in mangoes, and also plastic sorting system, and to identify drowsiness in drivers. All these aspects are being addressed in this Central Electronics Engineering Research Institute. And coming to NEERI Chennai Center, this is an Environmental Engineering Research Institute, and they work on the biomedical waste management as well as eco-friendly crackers, and also stone crushing, where the environmental pollution, all these aspects are being studied by them. So I would like to conclude with the Thirukural, Porul Karivi Kalam Vine Idanoda Aindum Irudira Ennichayal, which means we have to have perform our act after considering the 5 aspects: the resources, instrument, time, action, and context, so that our act is very impactful. And today we are very fortunate to have with us Professor Stephen Grill, who is the director of the Max Planck Institute of Molecular Cell Biology and Genetics, Germany. He is biophysicist by profession. Thank you, Professor, for your accepting our invitation and being with us today for our National Science Day celebration in spite of his your busy schedule. Thank you so much. And once again, I welcome you all for this function, and we are eager to hear from the chief guest his address. Thank you.
Thank you, ma'am. I request Dr. Anup, Chief Scientist, CSIR SCRC, to present a report on Dr. N. Lakshmanan Memorial Science Quiz, which was conducted as part of National Science Day celebrations.
Respected Chief Guest, Professor Stephen Grill, respected Director, and my dear colleagues, good evening to all. So I'll be presenting the report on the Dr. N. Lakshmanan Memorial Science Quiz conducted as a part of National Science Day celebrations in our campus on 28th February 2025. So this program was started from last year. Every year we are conducting this quiz program exclusively for the corporation schools in Chennai. We have another quiz program for all the schools as a part of our CSIR Foundation Day, but this is exclusive for the Chennai corporation schools, as this is an initiative started by our director, and this is in memory of our former director, Dr. N. Lakshmanan, who is a stalwart in the area of structural engineering. So this year we had participants from 31 schools. 93 students from 31 schools have participated in the quiz program on 28th February, and each school is permitted to have sent one team of 3 students, and the program was held in this hall, Wigan Auditorium, and the program started with a brief report on the organization of the program and the formalities by Dr. S. Maheshwaran, Senior Principal Scientist, CSI SCRC, and the quiz had 2 parts, a preliminary part and a final rounds, and the quiz was conducted by C. A.K. Fawaz Ahmad, our Principal Scientist, CSI SCRC, and a team of our young scientists. And in the screening test was a written test for half an hour, and based on the screening test, 6 teams were selected for the finals. And the finals consisted of several rounds, and in which all the 6 teams were actively participated, and it was very interesting, including the participation from the audience, and even the scientists of both CSI, SCRC, CMC, and the other staff members also participated in the event for the finals. So, and after the finals, we had a popular science talks by 2 of our scientists, Ms. Dr. Bharati Priya and Dr. Kirtana, to motivate these students on the science, popular science aspects. So that was also well received by the students. And the first prize for the quiz was won by team from CHSS Virumbakam, and second prize was won by CHSS Korattur, and the third prize was won by CHS Padikopam. In addition to that, all the other 3 finalists were given consolation prizes, that is CSH Rangarajapuram, CSH MGR Nagar, and CHSS Namalavapat. And the prizes were distributed by our beloved director on the same day to all the winners, and the consolation prizes also were distributed, and all the school teams were given participation certificates. So the program went on well with great success because of the contribution by number of people. The great number of people have helped us in conducting this event, including our own staff members, different divisions like SHRDD, BKMD. So I don't want to name all of them, and I thank all of them. And I also thank the Chennai Corporation Commissioner and the AEO for their help and support in conducting this event. Thank you all.
To explore new horizons and push scientific frontiers further, it is vital for research community to develop a passion for science. It is with this zeal we celebrate Science Day every year in our organization with guest lectures from eminent scientists and researchers. This year, we are fortunate to have Professor Dr. Stefan Grill, and to formally introduce him to the audience, I now request Dr. Saptarshi Sasmal, Chief Scientist, CSIR SCRC.
Respected Director, respected Chief Guest, Professor Stephen Grill, and my senior scientists and my friends, colleagues, students, and all, a very good afternoon to all of you. So it is my great pleasure to introduce Professor Stephen Greel, who is the director and the research group leader for this, the world-renowned institute of the Max Planck Institute for the Molecular Cell Biology and the Genetics. So if I have to go down the biodata, mainly the prizes and the accolades are a lot, but I will just tell you the brief to say that how the science day really fits so him for this, and we are so fortunate to have this time. Is the Professor Stephen Greel was born in 1974 in the Heidelberg and studied physics at the University of Heidelberg. He received his PhD from the European Molecular Biology Laboratory in Heidelberg and the University of Munich. He did his postdoctoral work first as the Max Planck Institute and then he continued in the University of Berkeley. Stephen Greel has returned to Dresden in the year of 2006, and where he worked jointly with the Max Planck Institute and the Max Planck Institute for the Physics for the Complex System in Dresden. He earned his habilitation in the theoretical physics from the University of Leipzig in 2013 and was a professor in the biophysics at the Biotechnology Center for the Technische University Dresden from 2013 to 2019. So he was the editor of the Journal of the Physics Review Letters from the year of 2014 to 2019. He was the founding speaker for the cluster of excellence for the physics of life. I think that we can also having the opportunity to listen from some part of that today at the University of Dresden from the 2018 to 2021, and became the director of the Max Planck Institute CBG from 2018. So where he and his group works to reveal the physical concepts and the principles that underlie actually dynamic self-organization of the living matter. So he has received a number of awards, actually, as I have mentioned you, but I just want to mention that two of them is that one is Elric and the Ludic, the Darmstadter Nachwuchs Prize. Suppose if I translate into English, it is a young talent award, and also the Raymond and the Beverly Sacker International Prize in the biophysics. Now, if I recall the lecture given by the director says that it is this year, this is the National Science Day, which has been focused on the empowering the Indian youth for the global leadership in the science and the innovation for the Vikshit Bharat. I think that Professor Gill would be the best fit for doing so. So with that brief introduction, I would like to invite Professor Gill for giving the National Science Day lecture. Please, Professor.
Thank you. Thank you for the kind invitation. Thank you for having me. And what I thought I would do in the next 25 minutes is give you a little bit of a glimpse of the science that we're interested in. And the science is concerned with the interface between the living world and the inanimate world. And I take it that many of you work in the inanimate world, as we've just heard from the director. So I hope that maybe in the next 10-15 minutes I can at least spark some excitement that maybe the living and the non-living world aren't so different, and that the concepts are perhaps similar at some level. And what I will do is I will tell you a little bit about the Max Planck Society and the institute where I come from, and then focus on exactly these types of concepts, and I will keep it very simple. I will not go into very complicated protein language. I will try to just illustrate to you the concepts and the questions, and I give you very few answers. Okay, so let's see if this works. Here we go. Okay, so I came here yesterday on a long flight from Dresden, which is in the east of Germany. You can see the red label here. And we have an institute that was founded 25 years ago in beautiful Dresden along the riverfront. It is snowy in Dresden until 2 weeks ago, so it is a little bit colder than it is here. Here's our institute in the sunset. We have 25 research groups, and we have about 250 to 300 scientists and a total staff of almost 450 people. And there are two buildings. There's the main building, which houses also the wet laboratories for doing life science research. This is the atrium. And then we have a theoretical computational institute, Center for Systems Biology, where we try to match the fields of biology with physics, mathematics, and the computational science. And we are a Max Planck Institute, and Max Planck Institutes are pure research institutes focused on big scientific questions and on the aim to build excellence in science at various places in Germany. As I was chatting before with the director, it's structured similar to your research institutes, which is that we have about 70 institutes, Max Planck Institutes in Dresden, and across the whole spectrum of science, but all are concerned with fundamental research. The Max Planck Society is about 75 years old, just last year. We are proud of our 32 Nobel Prizes. There were a few in just the past years. For example, Svante Pääbo for sequencing the Neanderthal genome from Leipzig, which is very close to Dresden, or our colleague Ferenc Krauss for achievements in quantum optics. This is a picture of Max Planck, and the vision of the society is, as I've stated before, is to do basic research at the forefront, driven by curiosity and focused on quality. And it's scientists discussing, scientists deciding about science. We follow the so-called Harnack principle, which is that people matter, not programs. So we try to get the best scientists into the society. We have flexible dynamic institutes. We are international across all levels. And we pursue a lot of interdisciplinary science, which I would like to highlight here in bold, because this is what I will spend the next 15 minutes perhaps on, is on the interface between the life sciences, and particular biology, and physics. And I will give you some introductory slides here now to illustrate the topic. And the question is, how do organisms form? How does an elephant form? How does fish form? Here's a football fish. How does a worm form? We work with this organism. It's a model organism. It's a nematode worm, Caenorhabditis elegans. It lives in the flower plots. It is about 0.5 millimeter long. It is about as thick as a hair. Okay, so it's a small organism. It is transparent. It can be frozen and it can be rethawed, which makes it very applicable to do research. We can generate genetically modified worm strains. We can freeze the worm strains. We can ship them. So it's just a very practical model organism, and it shares many of the complexities that we share. Of course, it's structurally a lot simpler than a human, but it has about as many genes as we do. It has much fewer cells, but the principles of how living systems structure themselves can be readily studied in this worm. So how do organisms form? This is really the question. How do cells and tissues form? We know we've learned a lot about the building blocks in the last 30 years, about the molecules and the chemistry in which molecules interact. But how many molecules come together to form a cell, and how cells come together to form a tissue? These are questions that are concerned with collective behaviors, of which we know a lot less than we do about the molecular scale. So here again, the worm, about 0.5 millimeter long. And what I will show you now, we will zoom in because we study how the embryo turns from a single cell into a whole-blown, full-grown organism. So we will zoom into the embryos. The embryos are inside eggs, which are stored inside the mother for some time. So we will just zoom in here. You see one such egg. It's about 50 micrometers long. So again, about as long as a hair's thick. It's really an egg-shaped object, 50 by 20 microns along the short axis. So it's really an egg-shaped, much softer than a chicken egg, but it's also surrounded by an eggshell. So this is what we can see when we put it under transmission light microscope. You can see the eggshell that surrounds the one-cell embryo. And what I will do now is play you a movie of how this one cell turns into almost everything that makes up the worm. So you see one cell that has in the center a so-called cell division apparatus that assembles. You can see the cell division apparatus move around and oscillate. Now the cell divides. We have two cells. Okay, now I play a lot quicker, 4, 8, 10 cells and more than 10 cells, division, division. You can see how the volume is divided up into smaller and smaller cells. Now we have more than 100. Now this mixture of cells will start to elongate itself in a process called gastrulation. You can see this a little bit. And you can see how it turns into a worm, into an elongated object. Okay, now the worm will start to crawl around inside the eggshell. It will leave the eggshell and go on to live its short life. The worms live about 20 days. But you can see, we can readily watch how a single cell that has the DNA, that has all the genetic information, turns into a full-blown worm over the course of just a couple of hours. So here's maybe a way to think about this. So what distinguishes this material from the materials that you here work with? I'm going to say that we are made of a special clay. This is from a talk that I gave at a children's university day, which is a little bit similar. It was a Saxonian a day to motivate children and high school children to move into science. So I made this little movie, or my son made this little movie. So excuse me, it's very simplistic, but I think it makes a point, because we are made of a special clay, but this clay can shape itself. Where do we have a material that can structure itself in the intricate ways that I've just shown you? Okay, so to make a full-blown worm. How does that work? What are the forces? Where do the forces come from that drive deformation? You are all engineers. You know that if I want to reshape an object, I need to apply a force. Where do these forces come from? What is the energy balance? How do I have to think about structure formation in these complicated? So how are the forces that drive these types of deformations that we see? These are the first questions that we started to work on. And not just us, about 15 years ago. This was also enabled by new types of microscopes. So a lot of progress in science comes from the development of new technologies, in particular better ways to image these types of processes. And I'm going to show you a movie now from zebrafish. Okay, so here you see lots of different cells labeled in different colors. I'm not going to tell you how exactly the labeling works, but what I would like you to take from this is that over the course of just about 6 hours, a circular bag of cells, so this is a fish, it's a lot closer to us, it's a much bigger organism. What you're seeing here is about 0.5 millimeter across. This is a spherical ball of cells that over the course of just 6 to 8 hours shapes itself into everything that essentially makes up a fish. I think you can see the backbone. You can see the backbone forming. You can see the eye lobes that form the brain. So if I were to play this movie another 12 hours, the fish would start swimming away. So how does that work? How can living matter structure itself into such intricate ways to form the most complex objects that we know of, which are living organisms that can walk around on their own and give a talk in front of a distinguished audience? So where do the forces come from? I will tell you a little bit more about this. They come from the surface of a cell. So I'm going back to the nematode worm. At the surface of the cell, there are little proteins. Proteins are the building blocks of life. And there are particular ones that we call molecular motors. They can burn a fuel. They can burn ATP, that is the energy battery of life. They can burn a chemical fuel so they consume energy to generate mechanical work. Okay? And they do this. This is best illustrated in a movie that is, of course, it's just an animation, but I think it takes a lot from. I will play it again. So here's one such molecular motor that walks along a particular track, and it can do work. It can pull. It can pursue. It can, generate forces. Move objects against opposing forces across distances. So really perform mechanical work. So this molecular motor, now I will show you a real movie. And in fact, the molecular motor that's relevant at the cell surface is essentially the same molecular motor that moves our muscles. Okay, it's called myosin. So here's my favorite movie of all times, which is an atomic force microscope, high-speed atomic force microscope movie. So now this is not an animation. This is a real movie where one such motor protein was recorded with a high-speed atomic force microscope, which is basically a needle that really quickly moves up and down and traces the shape. So these motors walk along a filament. You can see this helical track, and they walk like we walk in a head, it has 2 feet, so it doesn't let go in order to really move progressively. And per step, it consumes 25 kT, which is the energy provided by the hydrolysis of one ATP molecule. So this molecular activity that you can see here, amplified 1 billion times, is what gives rise to the structure formation processes that I've showed you, either in the worm or in the fish. And how does that work? Well, I'll give you a little bit more insight, and then I will end my presentation. So what you're seeing here is the tracks. Okay, so here, now let's look a little bit more at the cell surface. At the cell surface, there are the filaments along which the motors walk. They are represented along the surface in a kind of disordered fashion. And if we zoom in, here are the best microscopes that we have. We're looking at exactly the surface of the one-cell nematode worm. In red, you see the tracks, and in green, you see we have attached a little green fluorescent light bulb, if you wish. We use laser confocal fluorescent microscopy to take these images. So each green dot is one such motor. It's a collection, they assemble into units of about 10. So one such collection of motor. And you can see how this movie is just about 2 to 3 seconds long. It loops. We are watching a couple of microns across the cell surface. And the activity of the green motor proteins sets this fluid into motion. You can see that it slowly flows. The whole surface is slowly flowing from one side to the other. So this is basically a collection of molecular motors that operate in a disordered meshwork. And what we've been trying to do is understand the phenomena that arise when such motors operate together in particular collectives. What types of activities arise at larger scale? What is the emergent behavior, is the way to say this. And this was perhaps best cast by Erwin Schrödinger many years ago, prominent physicist, founding father of quantum mechanics, who, when he, towards the end of the Second World War, he was taken out of his usual environment. He worked at Trinity College in Dublin and started to think about matter at higher temperatures, not at ultra-cold temperatures where quantum phenomena are prevalent, and started to think about what makes living matter special. So he gave a lecture series, which is turned into a book, What is Life? I can highly recommend you to read it. It was written at a time where we didn't even know about the structure of DNA, yet it puts forward exactly the right questions concerning what it is that makes the state of living matter particular and different from inanimate states of matter. I'm reading you one sentence here. Living matter is likely to involve other laws of physics hitherto unknown, which, once revealed, will form just an integral part of science as the former. And the key thing is that living systems are driven out of equilibrium by energy sources at molecular scales, and this somehow leads to force generation, deformation, and movement, as I've showed you. And we're trying to understand how exactly that works. So this has, so now, 70, almost 80 years later, we can look at this problem again with a fresh mind. We now have fields of physics, a new field of physics. It's called, a field of active matter physics that is concerned with exactly answering these types of questions. A definition of active matter physics is perhaps soft condensed matter physics driven out of equilibrium at molecular scales. And these motor systems, the so-called cellular cytoskeleton, which I've introduced to you, is a prime example. You have meshworks of tracks on which these motor proteins operate. They burn a fuel, and they can generate forces in this network that turns into stresses at more macroscopic scales that can lead to deformations and so forth. So this is really a formalism for describing structure formation in living system, and we've been using this, we've been developing this formalism and been using it over the last 10, 15 years. We focused on 3 types of force generation processes. In 3D, this would be stresses. In 2D, we refer to them as tensions. We integrate stresses over the thin dimension. So these systems can generate isotropic active tension, contract equally in all sides. These filamentous networks can align. And when they align, then you can have contractions in one direction. You can have the stresses in the direction of alignment be larger than in the orthogonal direction, so you can generate anisotropic active tension. And what we've been studying a lot is also the ability of these systems to generate torques, because you could see that these filaments are helical. So torques can be generated, which can drive rotatory flows. So isotropic active tension generation generates flows. Anisotropic can generate contracting rings, which is important for dividing cells. And active torques can drive chiral events. And I will not say much more about that, but it's a core focus of our research, how that how chiral effects, for example, give rise to left-right symmetry breaking and place the heart on the left side. So how do they generate flows? Well, I give you another simple view. It's really not so complicated because these motors, they operate like spiders in a soccer net. I know you all play cricket here, but I have to show a soccer slide. So in some sense, the energy consumption of ATP by these molecular motors leads to force generation and movement and displacement, which will tighten up, which generates tension in this net-like structure, which will lead to a contraction of the cell surface, which can lead to flows and reshaping. And it's exactly these molecular motors that do this. So if this is the case, this is presumably what drives the flow. And if this is the case, then this network should be under mechanical tension. And I will show you now one experiment that we do a lot in the lab, which is to test for mechanical tension by cutting the structure. So if you cut the structure apart, and if it's under mechanical tension, you would expect, the bonds are broken, you would expect the structure to fly apart. So we can do exactly this by taking a pulsed UV laser to cut this very thin structure at the surface of a cell. So I'm showing you here that one experiment. It's not, the microscope doesn't image quite as nicely as the other one, but you will see in a second, here's the cut. And you can see how this meshwork flies apart after the cut. It seals, it regrows, there's huge dynamics, huge turnover, but this opening, and in particular the opening velocity, is indicative of tension, anisotropy and tension exactly along that direction. So the network is under tension, and if we now think about moving up, so these systems can generate isotropic active tension at the surface, these drive flows, and the last point I would like to make today is to move up another scale. Cells connect to form tissues. And the only point I would like to make here is that in the end, it's not so different. The cell surface, cells are connected to adhere to each other to form a surface of cells. So also in these surfaces of cells, there's little spider elements, force-generating elements. We refer to them as forced dipoles, active forced dipoles acting. There's many of them, and they can drive the reshaping of tissues as, for example, a fish would develop. And I'm showing you here, we haven't worked so much with the fish, but we've worked with a flower beetle, an insect, and here you can see a whole tissue reshaping itself in a process called gastrulation by exactly the same types of forces. So with this, there's an anisotropic tissue flow that is driven by force generation in the tissue, much as I've just illustrated. Or here is an example that we also work with, and this is basically the last thing I would like to show you. This is looking at bird development, avian development, where you see thousands of cells labeled in white forming a tissue where, again, over the course of just a couple of hours, this forms everything that makes up a bird. And what I am particularly fond of in the early steps here are essentially flat. The bird embryo develops on the egg yolk, which is essentially a two-dimensional surface. So living systems are highly complex, so it's nice to be able to reduce it to a quasi-two-dimensional problem for much of the development. That's why we're studying the system. And here again is the movie of the fish. So with this, living matter is dynamic. Living matter can structure itself into all kinds of shapes and forms, and living matter forms dynamic materials with unexplored properties. And active matter physics is the framework in which we can understand self-organization, structure formation in living systems, and to really get to the heart of how living systems structure themselves. So with this, maybe an answer to Erwin Schrödinger's question is, what is the new physics? Well, it is active matter physics, or the physics of life. And with this, I'll stop, and I'm happy to take some questions. Here's the group that I've been fortunate to work with over the past couple of years, and I'll thank you for your attention.
Any questions from the audience?
Please.
So, thank you, Professor, for the lecture. I hope I'm audible.
I can hear you, yes.
Yeah, so I want to know something like, so you talked about the formation of embryo, right?
Yes.
So what causes deformities in the embryos, maybe, if there is a defect in the embryo?
Yes.
So do you think the energy imbalance is the reason for causing those deformities? Because
Yeah, so there's many reasons, there's many processes that can go wrong when an embryo grows. And energy budget is one key issue. So the energy comes from the mitochondria inside the cell. This is the energy powerhouse that generates the ATP that these molecular motors burn. So if the energy metabolism is perturbed, if the energy flows aren't as they should be, this definitely gives rise to defects. But that's not the only reason. There's so many things that can go wrong when you generate such an intricate object that metabolism and also the forces aren't the first things. The genetic programs are themselves very complex. And as we know from medicine, also here lots of things can go wrong.
yeah, so were you able to reduce the dimension of that deformities also? Was the
So in some sense, so are we able to reduce the dimensionality of these types? So of course, forces are forces. So forces also provide an integrating element. In the end, you have a tension in a surface, or you have a bulk stress, and there's many things. So this is kind of an integrative element. So in the end, this is about reshaping an object. So exactly as you say, which if you reshape an object, the stresses, it's important to understand the stress and the force balance underlying this. And there's many things that feed into the active stresses that are generated. So this is exactly an integrative element that, that we're trying to really put a finger on. That, we have to understand the force balance also to understand these types of processes. And then later we can also look at what goes wrong if things go wrong, for example, in cancer. When objects grow much bigger than they should, then there's also forces involved.
Thank you.
Yes.
Energy can neither be created nor
Yes.
destroyed.
Yes.
So where is this source?
Yes. So where's the source of energy that drives this? So that is a particular. Of course, we eat, and that generates. That is metabolized into all the building blocks that drive the generation of ATP, for example. But here's a great problem that you're also pointing to, which is that an embryo cannot eat until the worm is done. Only when the worm is done can the embryo start metabolizing itself. So the oocyte and the zygote, the first cell, must contain all the energy that it needs to drive the structure formation process. So the energy balance, to study the energy balance, is also highly interesting. In fact, we have a research group at our institute that is concerned with exactly that type of question. But it's really transmission of energy. You turn in chemical energy, which is stored in that big cell, and into force generation, mechanical work to drive the reshaping. It's a very good point.
I have got one question.
Yes.
So before I put the question, first of all, so thank you so much for giving such a lecture, and because that we talk about the structural deformation,
Yeah.
not that you are talking of the deformation of the embryo.
Yeah.
So it is a really mind-boggling for us. So what I was understanding from you that motor protein and having a surface tension and which causing the
Yeah.
isotropic and anisotropic.
Yeah.
But how the system is maintaining a pattern?
Yeah.
Even for the anisotropy also, it is having a pattern.
Yeah. Yeah.
So how they
Yeah.
come in between?
So I'm very impressed you are asking all the right questions. And this is something I didn't talk about, but that we've spent a lot of time working on. And in the end, the thing that makes living matter also special is the multitude of different building blocks. We have 25,000 genes in our body. We have thousands of different types of proteins. So these need to move to particular places. And also the mechanical events help sorting and generating asymmetric distributions of particular proteins. So these surface flows that I could show you, they can either drive mechanical deformations like cell division, but they can also transport proteins and particular units that are important to sort them only into one daughter cell. And then this cell is different because it has a different mix-up of building blocks than the other cell. And then this is amplified in chemical ways. So there's a lot of chemistry that happens here as well. It's not just physics, it's also chemistry. But this is something where biology, of course, has made great strides. And the point of what I was trying to tell you here is that the mechanics is something that we're now catching up. Yes. Yeah. So so this is always a circle of many things coming together. But for example, many cells in our body, like the green surface of cells that I showed you at the end, they form two-dimensional objects, like our stomach. Okay, they're called epithelia, which means that they're a single layer of cells that forms a two-dimensional object. Okay, and those cells need to know on which side is the inside, where the food is, and on which side is the body, so that the cell starts to segregate all the proteins that chop up the food, the fantastic Indian food that I've been eating in the last couple of hours and days. These proteins digest the food, but they shouldn't digest the body. So the cell needs to know which side is which. And these are exactly these types of processes, also mechanical, that make a cell decide that each cell has its coordinate system, knows where it is, and understands. I'm anthropomorphizing. These are self-organization processes that dictate the cell, okay, here's where the digestive proteins have to go. Yeah. Yes. Yes. Yeah. 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 Exactly. Yeah, so those are the real, so I think for me that's also the biggest difference between the inanimate and the animate world. You saw how when I did the laser cut to measure tension, you saw that this was basically real time. So within 8 seconds, even though the material was flying apart because there's strong stresses in the surface, there's a huge surface tension, within about 10 seconds the material regrows. And it happens inside a cell, it also happens for a tissue. So, and there's an intricate balance of the systems self-organize to a kind of homeostatic state. If it's perturbed, there's stability, it'll go back to that state, but the rules by which this happens, we don't understand very much, to be honest. That would maybe the next National Science Day. But of course, the brain is perhaps one of the ultimate challenges that we have to understand. I will not say much about it here, but of course, the brain is made out of cells. Electrical communication becomes important in the brain. But I will say that my unbiased and inexperienced summary of the state in which I hope there's no neuroscientists in the audience, because I will say that I don't think we understand very much about the brain as it is at the moment. We're probably learning much more about it when we study ChatGPT. So, and maybe you can take that as a food for thought for an evening discussion. Thank you.
Thank you, Professor, for your spellbinding lecture and taking us through the life of dynamics of activities in the even tiniest of life forms. Of course, the true essence of science lies in questioning and curiosity. And the one from your institute has shown us very colorful and informative images and videos. On behalf of CSIR, SCRC, and CMC, we would like to express our heartfelt gratitude for your presence at this memorable occasion, and I request our director to present a memento to our chief guest. I now invite Dr. Harikrishna, Chief Scientist, CSIR, SCRC, to deliver the vote of thanks.
Good evening to one and all. It is my privilege and pleasure in proposing the vote of thanks for the National Day Function 2025. First of all, on behalf of CSIR SCRC, I thank the chief guest of this function, Professor Dr. Stefan Grill, Director, Max Planck Institute of Molecular Cell Biology and Genetics from Germany, for accepting our invitation and for delivering the National Science Day lecture on physics of life. I am sure every one of us are mesmerized by his presentation. I also like to thank our director, Dr. N. Anandavalli Madam, for giving the welcome address and also for providing her guidance and encouragement in organizing this event. I thank Dr. Parivallal, Advisor Management, for his suggestions in organizing this event. I thank Dr. Saptarshi Sasmal for introducing the chief guest of this function. And I also like to thank the Dr. N. Lakshmanan Memorial Quiz Competition Committee under Dr. Anup and his team for organizing the quiz competition exclusively for the students of Corporation of Chennai Schools on 28th February 2025 in this auditorium. I also thank all the 31 schools who have sent their students and the 93 students who participated in this quiz competition. I would like to thank the staff of Director Office and Advisor Management Office and various functional heads and their staff of BKMD, ICT, SHRDD, HRD, Works and Services, Horticulture, Electrical Section, Canteen, Guest House, AO, F&AO, and SNPO Security for their support in organizing this event successfully. At the end, I thank Dr. Kitna for comparing this event. I once again thank one and all who had helped directly or indirectly in organizing this event. Thank you.
That brings us to the end of today's function. I request all of you to please rise for the national anthem. Thank you.